Vaccines save lives. One of the things that we know is that there are many children who are alive today simply because of the institution of routine childhood immunization.
You know what? Vaccines are incredibly safe. Most side effects are very mild, like a little bit of fever or pain, and can be easily managed at home. More serious reactions, like allergic reactions, are incredibly rare.
So I think parents should be reassured that vaccines are safe and that their benefits clearly outweigh other risks.
Get your kids vaccinated.
Now, some people are saying kids who aren't vaccinated shouldn't be allowed to go to school or daycare or go to public places, but here's where I think it should go. If you are one of those people who knows more than doctors, that's fine, no vaccinations, but you're not allowed to go to the doctor anymore. Okay?
This is what I was talking about when I said that vaccine refusal is a symptom of a much larger problem. Because this culture of individualism is a crisis, and it affects most of the social problems we face as a society.
Baseless claims about a link between vaccines and developmental disorders have fueled the so-called anti-vaxxer movement for years now. But in Texas, the anti-vaxxers are motivated by something else, an unconditional commitment to personal freedom, even at the expense of public health.
Hey guys, it's your boy ZDoggMD. This is just a quick live I didn't intend to do, but this morning I was reminded of something. Anti-vaxxers, the cult of the anti-vax movement, like the professional delusional anti-vaxxers online who spend their time like trolling, are some of the stupidest, most concrete thinkers. It's like a third grade level intellect.
We haven't talked about the vaccine debate here on SciShow because there is no debate to have. Vaccines don't cause autism, and they save millions of lives every year.
My goal in this video is to introduce you to the debate that doesn't exist. I'm not a doctor, but I've been reading through the medical literature and research on this subject for years. It's very expansive. So I'm only going to look at one aspect, one question many vaccine-hesitant parents have. Is the aluminum used in vaccine adjuvants safe? Aluminum in its soluble form is ubiquitous to our environment. It's in the soil, in our water, in our food, in our cosmetics, in our medicines, and it's a known neurotoxin. Its free ion, Al3+, is highly biologically reactive and uniquely equipped to do damage to essential cellular neuronal biochemistry. It is linked to Alzheimer's disease, Parkinson's disease, multiple sclerosis, impaired renal function, muscle weakness, seizures, speech problems, and so on. Amazingly, out of all vaccine ingredients, only one has been studied for safety: thimerosal, a mercury preservative. This paper states, and I quote, "According to the U.S. Food and Drug Administration, safety assessments for vaccines have often not included appropriate toxicity studies because vaccines have not been viewed as inherently toxic. Another study states, in addition to antigens, vaccines contain small amounts of preservatives, adjuvants, and residual substances from the manufacturing process. Some parents have concerns about the safety of these ingredients, yet no large epidemiological studies have specifically examined associations between health outcomes and vaccine ingredients other than thimerosal. And from a 2018 study, FDA regulations require safety testing of constituent ingredients in drugs. With the exception of extraneous proteins, no component safety testing is required for vaccines or vaccine schedules. The dosing of aluminum in vaccines is based on the production of antibody titers, not safety science. So vaccine ingredients and health outcomes have not actually been properly studied due to the fact that vaccines and their ingredients are simply assumed to be safe. It's a bit of circular reasoning going on here. We don't need to study and test these ingredients because we've used them for years. And we've used them for years because they're assumed safe in the amounts we're using. I question this, and so do many doctors and scientists. As I was researching all of this the last few years, I came across a site called Vaccine Papers, vaccinepapers.org, and I was very impressed by the work that an anonymous researcher had done in collecting and analyzing some of the most relevant scientific data. So I don't know who to give the credit to, but many of the arguments and studies I'm now going to present here, I culled from his or her collected material and analyses. Aluminum has been used in vaccines since the 1930s. It's an adjuvant, commonly alhydrogel, used as an immune stimulator. In fact, what it does is it irritates the immune system a little bit to provoke a response so that the body will start producing antibodies to whatever it is the patient is being immunized to. What we're told is that we don't need to worry about the aluminum in vaccines because the body is going to excrete it quickly. The Oxford Vaccine Group states, after vaccination, there is a temporary increase in the amount of aluminum in the body, but this is not a lasting effect. The body gets rid of most of the aluminum in just a few days. There is no evidence that this causes any risk to babies and children. When it comes to the aluminum that we ingest from our food, 99.7% passes straight through our gut and is eliminated in feces. The other 0.3% is absorbed, carried into the bloodstream, and pretty quickly passes on through our urine. Most of it's gone within 24 hours. This is how the body protects us from heavy metal toxicity. This second chart from Vaccine Papers shows the way that vaccine advocates assumed excretion of aluminum from vaccine adjuvants happened. They thought it worked the same way as ingested aluminum. They believed that if you inject the aluminum into tissue, it will immediately dissolve into Al3+ particles, just like the soluble type of aluminum we ingest from our foods, and then be carried by the blood to the kidneys, which can then excrete it. But here's the problem. Water-soluble Al3 from aluminum lactate or aluminum chloride, which we ingest, does not behave the same way as the insoluble solid nanoparticle form of aluminum hydroxide, aluminum phosphate, or aluminum sulfate that is used in adjuvants. Because of the low solubility, most of the aluminum from adjuvants persists in the body, studies show, for a minimum of months and likely even years. A study was done to see how long it took for rabbits to excrete a radio-labeled aluminum adjuvant after injection. The radioactivity allowed them to take accurate measurements of content in the urine, blood, and tissues. After 28 days, they were dissected. The researchers found that only 6% of the aluminum hydroxide, or 22% of the aluminum phosphate adjuvant, had been eliminated from their bodies. The levels of aluminum in their blood, the levels of aluminum in their blood and urine mostly remained close to normal during this study because they weren't excreting very much. The adjuvant was detected in the brains, kidney, spleen, liver, lymph nodes, and heart. That's where the aluminum had traveled. It had gone to the organs and brain. In a study from 2015, they found that nanoparticles from aluminum adjuvant were still in organs and tissues of mice up to 270 days after injection. The authors stated, "The present study confirms that alum is extremely biopersistent and that alum, that's aluminum adjuvant, biopersistence can be observed in both the injected muscle and distant organs, including DLNs, that's draining lymph nodes, and spleen. Regarding the strong immunostimulatory effects of alum, long-term biopersistence of alum in lymphoid organs is clearly undesirable and may cast doubts on the exact level of long-term safety of alum-adjuvanted vaccines." In another study the next year by the same researchers, 1.3% of the total injected aluminum had made it into the brains of mice after 6 months. The MOVSAS study done in 2013 with infants showed that aluminum levels in their blood and urine hardly changed after vaccination. They tested them the day before and then the day after vaccination. The researchers said they were reassured to find no significant rise in serum aluminum levels after vaccination. But did they stop to consider what that means? It means the aluminum wasn't leaving the body. That should be concerning, not reassuring. Where was the aluminum? Macrophages are the body's garbage men. They find these little nanoparticles of aluminum at the injection site, and they'll try to eat them by destroying them with enzymes. The problem is, these insoluble nanoparticles are not digestible, so they just end up staying inside the macrophages. Most of the macrophages stay at the injection site, but some will travel through the body via the lymphatic system. And they will take those nanoparticles that they've picked up with them. These do eventually dissolve very slowly and go into the blood as Al3+, but in undetectable amounts. So mostly aluminum from the adjuvant does not appear in blood. This transportation of the nanoparticles in macrophages is so slow that it typically takes months or even years. They travel to the organs and the brain, causing damage wherever they go. So this chart on the Vaccine Papers site better illustrates what actually happens when the aluminum enters the body. As you can see, only a tiny bit is immediately excreted, and the rest persists for an undetermined length of time, only very slowly dissolving into Al3+ particles. The macrophages start moving around and crossing into the blood-brain barrier when there is inflammation in the body. It doesn't even have to be the brain, but that's when they really start moving their toxic load. It was a 2009 study that found that even liver inflammation caused macrophages to enter the brain and central nervous system. What happened is, inflammation caused microglia, that's the immune cells of the brain, to activate. Normally, they're in a resting state. An active state indicates inflammation, and what they do when activated is they attract the macrophages to the brain by releasing MCP1. That's macrophage chemoattractant protein, also known as CCL2. Unfortunately, like a Trojan horse, macrophages can bring some evil invaders when they're summoned. In a study by Kahn, nanoparticles were detected in the brain and spleen of patients up to a year after injection. The paper concludes, alum has high neurotoxic potential, and planning administration of continuously escalating doses of this poorly biodegradable adjuvant should be carefully evaluated by regulatory agencies, since the compound may be insidiously unsafe. It is likely that good tolerance to alum may be challenged by a variety of factors, including over-immunization, BBB, blood-brain barrier, immaturity, individual susceptibility factors, and aging that may be associated with both subtle BBB alterations and a progressive increase of CCL2 production. Remember, CCL2 is also known as MCP1, macrophage chemoattractant protein. MCP1 can be released by microglia by immune activation, including of just the sort that vaccines stimulate. Some infections or toxins induce MCP1. Interestingly, aluminum adjuvant induces MCP1, suggesting that it may stimulate its own transport. Do you see the significance of this? It means that nanoparticles can lie dormant in macrophages for years, and then when the person receives another vaccine, that immune activation causes the microglia to attract the macrophages, which causes them to start moving around and deposit their unwanted nasties. This may explain some of the damage from the MMR vaccine. MMR is given at 15 to 18 months of age, which is after aluminum-containing vaccines are given at 0, 2, 4, and 6 months. The measles vaccine can stimulate MCP1 production, and we know this from the 2014 study, a randomized trial of an early measles vaccine at 4.5 months of age, sex differential immunological effects. Therefore, the MMR vaccine may stimulate the movement of AANs received from prior vaccines into the brain. This may explain how MMR could cause aluminum toxicity even though it does not contain aluminum adjuvant. AANs refers to the nanoparticles from aluminum adjuvants. Now, is MCP1 elevated in the autistic brain? Yes. That's what a 2005 Vargas study found. It's the same thing a 2017 study found. And MCP1 was found to be elevated in newborns 24 to 48 hours after birth that later became autistic. The babies with elevated MCP1 will experience transport of aluminum adjuvant into the brain after vaccination. So the fact that MCP1 is elevated in babies later diagnosed with autism supports a causal link between aluminum adjuvant and autism. So just how much aluminum is needed to cause inflammation of brain tissue? Concentrations of aluminum as low as 10 to 100 nanomolar can cause inflammation of brain tissue. 10 nanomolar is 270 nanograms of aluminum per liter. That's an amazingly low concentration. A typical 1-year-old infant has a brain weight of about 1,000 grams. And 10 nanomolar concentration in 1,000 grams contains 270 nanograms. This is extraordinary because a single vaccine can contain 250 micrograms or 250,000 nanograms. And an infant can receive about 3,675 micrograms in the first 6 months. In other words, less than 1/100% of the aluminum in the first 6 months of vaccines can create a 10 nanomolar concentration in the brain. And 0.1% can create a 100 nanomolar concentration in the brain. A single vaccine contains more than enough aluminum to inflame the brain. Of course, this assumes uniform distribution of the aluminum in the brain, which is not the reality. Aluminum concentration in the brain is highly non-uniform. However, the above calculation establishes plausibility. The amount of aluminum in vaccines greatly exceeds the amount necessary to cause brain inflammation. End quote. So no, the aluminum from vaccines is not eliminated in a matter of days. It persists long-term and can travel to the organs and brain, causing inflammation. And it doesn't take much. I mentioned a study a few minutes ago in which 6 months post-injection, some of the aluminum had traveled into the brain of mice. They studied 4 groups of mice. One group was injected with 200 micrograms per kilogram of body weight of aluminum adjuvant, one with 400 micrograms per kilogram of body weight, and one with 800 micrograms per kilogram. The 4th group was control. Then they were subjected to a number of tests to find out how it may have affected them. You can see how they turned out in this chart. In an open field test, the mice are placed individually in an empty walled enclosure and allowed to explore. The movements of the mice are tracked by computer vision, which measures distance traveled and time spent in different areas of the field, and other characteristics. Open field tests are well known and are used to detect neurotoxicity. Open field tests have been used in neuroscience for decades. Look how poorly the 200 microgram per kilogram of body weight group performed. The changes indicate that this group experienced neurotoxic damage. It was also found that their brain aluminum content had increased 50-fold. 1.3% of the total injected aluminum had made it into the brain. The other 2 groups, who had received higher injections, were not significantly affected. It also caused microglial activation. Remember that microglia are the immune system cells of the brain? Vaccine Papers explains, activated microglia are capable of producing interleukin-6, that's IL-6, and interleukin-17, cytokines that cause autistic behavior in animals. Activated microglia play a role in autism. Human autistics have chronic microglial activation throughout the brain. The 2015 study, Role of Microglia in Autism, Recent Advances, looks at this. Now, why did the 200 microgram group fare the worst? This seems counterintuitive. Here's what appears to have happened. At the higher dosages, the body formed a granuloma, a hard nodule in tissue produced in response to injury. Because of intense inflammation at the site, it walled off the threat. That contained the aluminum and prevented it from traveling around. At lower dosages, the mice weren't so lucky. The body didn't respond by making a granuloma, so the nanoparticles were free to travel around the body. Here's the disturbing part. Of the 3 dosages, the 200 microgram per kilogram of body weight dosage is the one closest to the levels found in vaccines. Look at this comparison. A study was done to see how often granulomas form in infants in response to vaccines. Results from the paper state, the demonstration of long-lasting itching vaccination granulomas in 38 of 4,558 children who received aluminum-adsorbed vaccines during their first year of life confirmed our hypothesis that 0.10 to 1% of the children in the study would be afflicted. So this could mean about 99% of vaccinated children have the aluminum still traveling in their body, just like the 200 microgram per kilogram dosed mice. Higher dosages may induce a granuloma or reduce macrophage mobility, but at lower dosages, macrophages laden with toxic nanoparticles are more likely to move through the body. So multiple small injections of aluminum adjuvant, much like you would get following the vaccine schedule, may be more harmful than a single large injection. The dose makes the poison rule of classical chemical toxicity appears overly simplistic where it concerns aluminum adjuvants. Dr. Chris Exley, who has a PhD in the ecotoxicology of aluminum, has researched the subject for decades. He did not at first believe that there was any correlation between aluminum in vaccines and autism, but what he found over the course of his research changed his mind. In 2017, he said he believes it does play a role. He took measurements of aluminum levels in autistic brains for the first time. He took a 1 gram brain sample from each of 4 lobes, the occipital, frontal, temporal, and parietal, in the autistic brains. A couple of very interesting things turned up from this study. Aluminum levels were alarmingly high in their brains. The levels varied enormously, even in different parts of the same brain, which demonstrates that aluminum distribution is non-uniform. It's very unevenly spread. And the majority of aluminum was identified in non-neuronal cells, including microglia and astrocytes. Dr. Exley explains, there was clear evidence of inflammatory cells heavily loaded with aluminum entering the brain via the meningeal membranes and the blood-brain barrier. The fact that the majority of aluminum found in brain tissues in autism spectrum disorders was intracellular and associated with non-neuronal cells is, at least for now, unique to ASD and may begin to explain why young adolescents had so much aluminum in their brains. Now, his study has been criticized for lacking controls, but he explains that he was unable to find true matching controls, but demonstrates from his previous decades of research that he has plenty of data from other brains he has studied to be able to make that comparison and basically say, oh yeah, what we're seeing is different. This is so much higher than what we normally see. There's a really good article on Vaccine Papers further examining the limitations of Dr. Exley's paper, but also presenting solid contextual evidence that would support Dr. Exley's findings as accurate and highly significant. So if you have any questions concerning the validity of the study data, I would encourage you to read that article, Autistic Brains Have High Aluminum Levels.
So we won't get too much deeper into Alzheimer's, except that obviously you came to a very strong conclusion that there was a strong connection between aluminum and
well, I came to the conclusion that no aluminium, no Alzheimer's.
No aluminum, no Alzheimer's.
In the brain.
Wow. So then you decided to look at autism. You said that you were skeptical, that you didn't see that.
As a scientist, you clearly read the literature, stuff that other people produce. There were a number of papers making the suggestions. I wasn't convinced neither by the quality of the papers and the research, but even then I have to think about, so what is the mechanism? That's what I learned just working on fish. What is the mechanism? You must have a mechanism as well as just something being there.
Right.
And I couldn't think of a mechanism. But by going to the brain tissue, it would at least give us an opportunity to make the first major step. Either we find no aluminium beyond the method blank type value in the brain tissue of people with a diagnosis of autism, or we find some aluminium, or we find a lot of aluminium.
What did you find?
We found a lot of aluminium. We found a scary amount of aluminium.
Interleukin-6, IL-6, is a cytokine, as I said before, and part of the immune system that plays an important role in brain development. But elevated levels in the brain during early development have been proven to cause autistic behavior in animal experience. An elevated IL-6 is found in autistic patients. Now, correlation doesn't equal causation, but there's more. A study was specifically done to see if autism-like behavior is caused by immune activation. And the results showed that IL-6 was necessary and sufficient for autistic behavior. Other studies have confirmed this. All the studies just mentioned looked at maternal immune activation. In other words, the fetuses were exposed to IL-6 in the womb by stimulating immune activation in the mother. But in another study, a virus carrying the IL-6 gene was injected into the brains of mice after birth. Control mice were injected with the same virus, but lacking the IL-6 gene. The paper stated, "We demonstrate that IL-6 is an important mediator of autism-like behaviors. Mice with an elevated IL-6 in brain developed autism-like behaviors, including impaired cognition ability, deficits in learning, abnormal anxiety-like trait and habituation, as well as a decreased social interaction initiated at later stages. These findings suggest that an IL-6 elevation in the brain could modulate certain pathological alterations and contribute to the development of autism. What this all suggests is that if a woman experiences an elevated immune response during pregnancy, it may lead to a neurodevelopmental disorder in her baby, such as schizophrenia or autism. Cytokines are part of our immune system, and their levels increase in our blood when we have an infection or when our immune system is activated. What can happen in babies whose mothers experience an elevated immune response during pregnancy is that these IL-6 cytokines may produce a chronic, slow burn inflammation in the brain, which may eventually present as autism in the child. I'm going to point something out here. The possibility of a vaccine during pregnancy being the perfect trigger is strongly plausible. The vaccine's very purpose, specifically the aluminum adjuvant used in it, is to induce an elevated immune response in the mother's body. But this can have unfortunate results for the baby. We also know that aluminum increases IL-6 in the brain. This study here looks at the role of selenium in counteracting the toxic effects of aluminum, raising IL-6 levels. Short-term exposure, 16 hours, to aluminum resulted in an increase in the systemic inflammation parameters IL-6 and PAI-1. Both the increase in IL-6 and decrease in glutathione status could be prevented by co-exposure to selenium. The more times the immune system is activated, through vaccinations, for example, the more sensitive and reactive it may become. So if a mother gets vaccinated while pregnant and then vaccinates her baby when it is born, the more times the child's immune system is irritated in this way, the higher the chances it may result in chronic inflammation, which could lead to autism. It is known that the brains of autistic patients are inflamed and they have a heightened immune response. We don't have time here to examine the scientific evidence regarding the link between vaccine adjuvants and autoimmunity. That's a whole trail of its own. But a good case can be made from the medical literature that adjuvants like aluminum are playing a role in the rise of autoimmune disorders. In fact, there's a new term for it in the medical literature. In 2011, a new syndrome termed ASIA, that's autoimmune or inflammatory syndrome induced by adjuvants, was defined, pointing to summarize for the first time the spectrum of immune-mediated diseases triggered by an adjuvant stimulus, such as chronic exposure to silicone, tetramethylpentadecane, pristane, aluminum, and other adjuvants, as well as infectious components that also may have an adjuvant effect. All these environmental factors have been found to induce autoimmunity by themselves, both in animal models and in humans. The data herein illustrate the critical role of environmental factors in the induction of autoimmunity. Indeed, it is the interplay of genetic susceptibility and the environment that is the major player for the initiation of breach of tolerance. Experimental evidence also shows that simultaneous administration of as little as 2 to 3 immune adjuvants can overcome genetic resistance to autoimmunity. Aluminum is an experimentally demonstrated neurotoxin and the most commonly used vaccine adjuvant. Despite almost 90 years of widespread use of aluminum adjuvants, medical science's understanding about their mechanisms of action is still remarkably poor. There is also a concerning scarcity of data on toxicology and pharmacokinetics of these compounds. In spite of this, the notion that aluminum in vaccines is safe appears to be widely accepted. Experimental research, however, clearly shows that aluminum adjuvants have a potential to induce serious immunological disorders in humans. In particular, Aluminum in adjuvant form carries a risk for autoimmunity, long-term brain inflammation and associated neurological complications, and may thus have profound and widespread adverse health consequences. In our opinion, the possibility that vaccine benefits may have been overrated and the risk of potential adverse effects underestimated has not been rigorously evaluated in the medical and scientific community. We hope the present paper will provide a framework for a much needed and long overdue assessment of this highly contentious medical issue. More and more researchers are coming to understand the significance of the interplay between genetic susceptibility and environmental triggers. And there's a term in medical literature for that now. It's called GXE, which refers to gene-environment interactions. Even if your genetics are a loaded gun, it may still not fire until one or more environmental factors pull the trigger. In a 2015 study, the author stated, both human and animal studies suggest that GXE plays a role in ASD pathogenesis. And again, compared with individuals without autism spectrum disorder, brains of individuals with ASD demonstrated downregulation of 209 genes enriched for gene categories related to synaptic function, whereas 235 genes implicated in immune and inflammatory response were upregulated. The former group was significantly enriched for association signals in genome-wide association studies, which is a research technique in which people are scanned for genetic variations, which are then tested for statistical associations with health outcomes. Whereas the latter group was not. These studies suggest that relevant immunologic changes are likely caused by environmental factors. Some quotes from a few more studies. These studies have demonstrated that genetic predispositions for a number of psychiatric disorders interact with environmental influences to manifest disorder. Gene X environment interactions play critical roles in the emergence of autism and schizophrenia pathophysiology. Multiple lines of evidence suggest that the roles of genetic and environmental factors depend on each other. Gene-environment interactions may underlie the paradox of strong environmental factors for highly heritable disorders, the low estimates of shared environmental influences in twin studies of serious mental illness, and the heritability gap between twin and molecular heritability estimates. Vaccination is an environmental factor. Vaccines obviously interact with the immune system. A genetically vulnerable immune system may produce abnormally strong or persistent inflammation in response to vaccination or vaccine aluminum adjuvant. So why, when 100 children receive vaccines, do only 1 or 2 of them become autistic? It appears to be based on a wide range of variables for the individual, including both genetic and environmental factors. If everything aligns right, or shall we say wrong, the brain becomes inflamed, and the child subsequently receives an ASD diagnosis. But it's more than a mere 1 or 2% who are being harmed, because neurobehavioral disorders and neurodevelopmental delays encompass a wider range than an ASD diagnosis, to say nothing of autoimmune disorders, seizures, and a host of other problems associated with aluminum toxicity. The 2018 Karowski study is widely cited by vaccine advocates, and really it represents about the best evidence they have that aluminum adjuvants do not cause harm. However, the study is fatally flawed. So Karowski took 85 infants between 9 and 13 months of age and collected the following data for each one: the total lifetime dose of aluminum adjuvant they'd received from vaccines, the aluminum levels in their hair and blood, and the Bayley scale of infant and toddler development scores, which is a measure of cognitive, language, and motor development. Then he looked for a correlation between 1 and 2 and between 2 and 3. He found none. So he triumphantly concluded that there's no correlation between vaccine adjuvants and aluminum toxicity. But what have we learned now about aluminum levels in hair and blood? We saw that because aluminum from adjuvants are insoluble nanoparticles, they don't dissolve in the blood like the soluble Al3+. Neither do their levels correlate with what's in the hair, according to the Bohr 2014 study. So of course he wasn't going to find elevated blood levels of aluminum. He failed to investigate a third possibility, a correlation between 1 and 3 lifetime aluminum adjuvant dose and the BSID score. That's what he should have examined. Point 2 is going to tell him nothing. It was pointless for drawing conclusions from. A third problem is that not enough time had elapsed from the point that the infants received their vaccines to the period of testing. There wasn't a sufficient amount of time for the aluminum from all the vaccines to make it into the nervous system. Neurodevelopmental disorders aren't usually detected at that age. Autism is generally diagnosed when the child's 3 to 4 years old. If you look at this chart on vaccine papers, development trajectories aren't very different between autistic and unimpaired children at the age of 9 to 13 months. So again, he couldn't have expected to find much. And then he had such a small number of participants. Remember that autism only affects 1 or 2 children out of 100. So really, he would have needed hundreds more subjects to be able to detect autism. You know, it's almost like this study was designed not to find anything. Maybe it was just sloppy oversight, but it's hard to understand how you could get so much wrong. The study was nonsensical. It doesn't provide us with scientific evidence that aluminum adjuvants are safe. The FDA MITCUS study is the other heavyweight evidence that vaccine promoters rely on. In a nutshell, what the MITCUS study did was compare the aluminum from diet and vaccines to a safety benchmark called the minimum risk level. With the MRL, it's considered safe to ingest a certain amount of aluminum a day without health effects. So then they calculated the aluminum burden on the body from vaccines. The problem here was the aluminum adjuvant was not considered part of the body burden as long as it was in its solid particulate form. Only dissolved Al3+ ions released by slowly dissolving adjuvant were included in what this study considered a burden. And finally, when they had these measurements of what the body was dissolving and excreting, they compared it to the MRL. And since what was leaving their body from the vaccines was less than the daily amount it's considered safe to ingest, they declared the aluminum safe. Yay! But we've examined the reality that very little injected aluminum leaves the body. It takes a long time to get rid of it. So one of the primary problems with this study is that it ignores the toxicity of those nanoparticles that aren't leaving the body. It didn't even count them in its body burden count. It erroneously assumed that only the dissolved Al3+ is toxic. But adjuvant nanoparticles are not harmless. They're biologically active and highly toxic. Their surfaces are chemically reactive and catalytic. Another major flaw for the study is that the MRL, the minimum risk level, was calculated from a feeding experiment with water-soluble aluminum lactate, not insoluble and persistent aluminum adjuvant particles. The safety of injected aluminum adjuvant can only be proven by experiments with injected aluminum adjuvant, which comprise insoluble and persistent particles. Studies of ingested water-soluble aluminum are not a good substitute. Scientific studies have established that injected aluminum adjuvant has unique toxic properties and ways of moving around the body, kinetics, that are not the same as water-soluble aluminum. You can read a 2017 scholarly critique of the fatally flawed MITCUS study that was published in the Journal of Inorganic Biochemistry titled Critical Analysis of Reference Studies on the Toxicokinetics of Aluminum-Based Adjuvants. So again, they're failed to prove anything. Dr. Paul Offit is a vaccine advocate and a doctor at Children's Hospital of Philadelphia. The rotavirus he developed made him a lot of money. So he has some conflicts of interest going when he comes out and tells everyone that vaccines are absolutely safe. He made the outrageous assertion that aluminum is a nutrient, a mineral with healthful functions in the human body. His statements on this have actually been removed from the Children's Hospital of Philadelphia website, but here's how he reasoned through this. He said, aluminum quantities fluctuate naturally during normal cellular activity. It is found in all tissues and is also believed to play an important role in the development of a healthy fetus. He pointed out that during healthy pregnancies, the amount of aluminum in a woman's blood increases, hence it must be a good thing. Vaccine Papers responds, what is actually happening with aluminum blood levels during pregnancy is that aluminum is being mobilized from the bone. The bone is a storage site for aluminum, just as it is for lead. During pregnancy, minerals are drawn out of bones to be used by the developing fetus. This is well known. Consequently, toxic metals stored in the bone are released as well. Both aluminum and lead are released by bone demineralization during pregnancy. This doesn't mean that either aluminum or lead are safe and healthy. The only role they play in pregnancy is damaging the fetus. Dr. Offit further cited the fact that preemies have more aluminum in their blood and that their mothers have more aluminum in their breast milk as evidence that aluminum is safe. How on earth could you come to this conclusion? The heavy metal toxicity in their blood likely played a role in causing the preterm birth. Dr. Offit deceptively cites the 1986 Ganrot paper on aluminum toxicity to bolster his argument that aluminum is not harmful. But the paper he appeals to demonstrates just the opposite. The paper concludes that aluminum is present in very small amounts in living organisms, but is abundant in the environment. In no case has aluminum been shown to have a definite biological function. Taken together, this suggests that aluminum possesses properties incompatible with fundamental life processes. Despite this, aluminum has generally been regarded as virtually biologically inert, and the interest shown for its biochemistry and metabolism has been very limited. However, during recent years, an increasing number of toxic effects have been established. So, Dr. Offit misrepresents the paper, which does not support his position. In 2003, Dr. Offit and a co-author published the paper, Addressing Parents' Concerns. Do vaccines contain harmful preservatives, adjuvants, additives, or residuals? In the section on aluminum adjuvants, he stated that in the Golub study, no adverse reactions were observed when mice were fed quantities of aluminum as high as 62 milligrams per kilogram of body weight a day. In reality, the data shows they did suffer fur loss and a cyclic pattern of food intake, a known sign of toxicity. Golub said their food intake pattern was similar to that seen in rodents eating diets deficient in a specific nutrient or adulterated with a poison. That poison was aluminum. It seems Dr. Offit has a difficult time telling the truth. Vaccine critics have repeatedly pointed out the lack of studies in the health outcomes between fully vaccinated and zero vaccinated children. And vaccine advocates have repeatedly responded that randomization is unethical. You can't withhold treatment, in this case vaccines, from the subject. So it's not unethical to recommend every planet on the child have all these vaccines. But it is unethical to conduct randomized trials comparing outcomes in order to get a better understanding of the vaccine safety profile so that we can know whether we ought to be recommending these to every child on the planet. That's the logic. They will also say, observational studies are useless because of selection bias. Almost all studies of vaccines are observational and therefore vulnerable to selection bias. So this is a hypocritical argument. And finally, they'll say, but we already have vaxxed versus unvaxxed studies, and they show that vaccines are safe. So Vaccine Papers examines 3 studies cited by vaccine advocates, but first defines the necessary criteria we need to see in a study of this sort, which includes a never vaccinated control group. This control group has never received any vaccines. It would also need to include a fully vaccinated exposed group. This group must receive all or nearly all the vaccines in the CDC schedule. The study needs to measure long-term health outcomes, especially neurological and immune-related outcomes, such as autism, mental illness, learning disabilities, allergies, autoimmune disorders, and the like. The follow-up period should be at least a couple of years because it takes this long for these health conditions to appear. And the study needs to have enough subjects to have sufficient statistical power to detect adverse outcomes at about the 1 to 2% level. So this will require a few hundred subjects per group. Here's a little chart that's been shared. No studies comparing general health of vaccinated versus unvaccinated? What about these? The last 2 studies Vaccine Papers did not address because they were limited to the flu vaccine alone, and their study results, I quote, are almost certainly explained by healthy user bias, end quote. But the first 3 are relevant, so they're addressed one by one. In this first study, Thoughts Capism made a mistake on the number of involved participants. The study actually included 13,453 subjects, not 18,000. But that's somewhat beside the point. The study looked at 13,359 subjects who had received vaccines and only 94 who had had no documented vaccination. So though the overall number of participants makes it appear that this study has high statistical power, it actually doesn't. There's a big imbalance in the group sizes. Anyway, the study went on to measure adverse outcomes such as respiratory allergy, eczema, asthma, and vaccine-preventable diseases like measles, mumps, and so on. These are some of the problems with the study. Neurological and autoimmune disorders were not measured at all. They didn't even look at them. And that's a major flaw in a study of this sort because these are 2 of the things we're most concerned about, especially with aluminum adjuvants. Allergic disorders can take years to appear, and some vaccines are given after age 1, so the 1 to 5 age group couldn't have shown us enough either. The vaccinated group is defined as receiving at least 1 vaccine. That means that group is diluted with only lightly vaccinated individuals, because you only had to have 1 vaccine in order to be considered vaccinated. So if we're comparing someone who's only had 1 vaccine to someone who has had 16 vaccines, that's kind of apples to oranges there. The vaccinated group received an unknown number of vaccines, and many may have had low vaccine utilization as compared to the CDC schedule in the US. Because of a number of serious problems and weaknesses, this study fails to tell us much about long-term vaccine safety outcomes. The second German study from that chart looked at the risk factors for asthma. Vaccine exposure was one of many exposures considered. Vaccines weren't even the focus of the study, which had a long follow-up of 20 years. There was no zero vaccine group. The study completely ignored all vaccines other than MMR, BCG, and TE. Since only MMR, BCG, and TE vaccines were considered, the unvaccinated actually received vaccines, just not the 3 specific ones that were being studied. Again, neurological and autoimmune outcomes were not measured, and total vaccine exposure was not described. Both vaccinated and unvaccinated groups received an unknown number of vaccines. So again, this paper is completely irrelevant to the present discussion since it's not even a vax versus unvaxxed study. It was actually comparing vaccinated children to other vaccinated children. Finally, the Philippines study. Of the 3, this is the only study here, and possibly one of the only studies at all that actually compared zero vaccinated children to fully vaccinated children. So it did meet the first and second criteria. It looked at cognitive performance, verbal, math, and language test scores in 10-year-old children in relation to vaccines received at 0 to 2 years of age. Subjects were born in 1983 or 1984 and follow-up cognitive testing was done in 1994. The study was published in an economics journal, which explains why it did not look at any health or disease outcomes. But of course, cognitive performance is generally related to neurological health. It had 1,022 zero-vaccinated participants and 85 fully vaccinated participants. Here are the problems. At the time of the study, only a few vaccines were being given, and only one, the DPT, contained aluminum. And by the study criteria, only one dose of it was necessary to be considered fully vaccinated, along with polio, measles, and tuberculosis. Trying to compare this to the CDC schedule of vaccines, the amount of aluminum children receive today, is disingenuous. Today, they receive 11 doses of aluminum-containing vaccines by age 6 months and 16 doses by 2 years. Comparing it by upper microgram limits, 625 versus 3,675 in the first 6 months. Also at that time, the polio, measles, and TB, BCG, vaccines were live attenuated and without adjuvant. The live vaccines create an immune response more similar to natural infection than aluminum adjuvanted vaccines. They generally produce a Th1 type immune response. This is relevant because early life and Th1 stimulation seems to have beneficial effects on brain development. Natural infections, which often stimulate Th1, improve a baby's brain development. The Th2 activation impairs brain development and causes allergic disorders. New research shows that BCG vaccine, live, stimulates Th1, improves brain development, and Hep B vaccine, which contains the aluminum adjuvant and stimulates Th2, impairs brain development. So it is biologically plausible for the polio, measles, and tuberculosis vaccines to improve cognitive ability. But this is not relevant to the US CDC vaccine schedule, which contains many more aluminum adjuvanted vaccines. And the CDC schedule no longer includes TB, live polio, or the measles vaccine by itself. It's combined in MMR. There's also the possibility that selection bias may account for the results in that study. For example, the vaccinated had mothers with more education, higher socioeconomic status, they lived in larger homes, had more toilet and sanitation access, had fewer siblings, had better nourished mothers, and had more breastfeeding as infants compared to the unvaccinated. So there's that too, which may have given their cognitive abilities a boost. And again, there were far too few participants on the vaccinated side. The study is irrelevant to the discussion if we're looking for things like autism, which isn't even necessarily going to show up once in a group that small. Only one study has been done that has actually technically met the criteria that vaccine papers listed, comparing hundreds of partially or completely vaxxed children to hundreds of completely unvaxxed children and measured health outcomes dealing with neurodevelopmental disorders, autism, learning disabilities, and so on. This peer-reviewed study that looked at a total of 666 homeschooled children between the ages of 6 and 12. 261 were vaccinated and 405 were partially or fully vaccinated. And it found that the vaccinated were almost 4 times more likely to be diagnosed with neurodevelopmental disorders, over 4 times more likely to be diagnosed with autism and ADHD, over 5 times more likely to be diagnosed with learning disabilities, and over 30 times more likely to be diagnosed with hay fever allergies. The weakness of this study was that the researchers did not have access to medical records. Instead, they relied on the use of an anonymous survey. So, more research really needs to be done. But vaccine advocates don't seem interested in doing the kind of study that would truly compare neurodevelopmental and immune disorders between the fully vaccinated and zero vaccinated. Do any of these studies by themselves prove that vaccines cause autism and neurodevelopmental disorders? No. But taken together, they do establish biological plausibility. This is significant and demands more research. Is it possible that we're trading the risk of acute illness for the risk of chronic illness? I'm not arguing here that vaccines just don't work. I'm asking, is our current risk-benefit analysis assessment accurate? Could it possibly be that there's actually more risk than we've been told? Vaccine advocates often imply that the safety of vaccines is just an open and shut case. But they're either unaware of the science that we have available today, or they're ignoring it. Far from putting all questions to rest, the medical literature on the subject continues to raise them. Yet many of them act like anyone who questions vaccines is an IQ-challenged Neanderthal, or even worse, a child abuser. More and more, the discussion of mandated vaccinations is being placed on the table. And researchers are continuing to develop aluminum adjuvanted vaccines, which will likely be added to the schedule at some point. So we need to be able to have an honest discussion in the open in a hurry. The fact that you can't discuss this openly is a big red flag. It will get you censored, and if you're a doctor, it could put your career in jeopardy. I might point out that the people and organizations who are saying there's no connection between COVID-19 vaccine and heart attacks are the same people and organizations who have said there's no connection between neurological disorders and vaccines. It would take a whole video presentation of its own to even scratch the surface of the corruption and fraud which has been uncovered among not just one, but all manufacturers of childhood vaccines, as well as all the related government regulatory agencies. Dr. William Thompson, a CDC senior vaccine safety scientist turned whistleblower, came forward with the receipts, lots of documents and audio files, demonstrating dishonesty and corruption within the CDC. While Dr. Thompson is a vaccine advocate, he testified that he and his co-authors at the CDC intentionally omitted data from one of their studies that demonstrated a causal link between the MMR vaccine and autism.
Here's the deal: is the CDC is they're paralyzed. The whole system is paralyzed right now, and the whole branch is paralyzed, and it's becoming more paralyzed. So there's less and less and less being done as the place just comes to a grinding halt. So really what it needs is, for Congress just to come in and, say, give us the data and we're going to have an independent contractor do it and bring in the autism advocates and have them intimately involved in the studies. When I talk to you, you have a son with autism, and I have great shame now when I meet families with kids with autism, because I have been part of the problem. I shoulder that the CDC has put the research 10 years behind, all right? Because the CDC has not been transparent, we've missed 10 years of research because the CDC is so paralyzed right now by anything related to autism. Right. They're not doing what they should be doing. Right. They're afraid to look for things that might be associated. I'm completely ashamed of what I did.
Parents are not only justified in asking questions, they have a responsibility to do so. Don't outsource your thinking to others, not even experts. Make your own informed choices for yourself and your family.